Hello? 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 Last class we completed water potential and observation water. Absorption of water and movement of water. Ascent absorb. Through ascent absorb, finally the water reaches stomata. What happened through the stomata? After reach the stomata, from the stomata they can evaporate. This process called as, hello? Hello?
Oh, yes, sir.
Yes, is it audible to you?
Yes, sir.
Right. So the process called as transpiration. Transpiration is defined as the loss of water in the form of vapor from the living tissue of aerial parts such as leaves and also tender stems, so on. So the transpiration process also based on the process of osmosis. We know osmosis, through osmosis, absorption process, and movement of water and transpiration of water, right? So let's see the mechanism of mechanism of stomatal movement. That means opening and closing of stomata. So before that, we know the stomata structure. If we see stomata in dicot, it is bean shaped. Let me share the diagram. Are you able to see this diagram? Hello?
No, sir. Yes, sir. Yes.
Right. So these are the guard cell, this bean shaped structure. This is guard cell and surrounded by subsidiary cells. Surrounded by subsidiary cells, right? Guard cells. And surrounded cells, subsidiary cells, right? So guard cells, already told you, bean shaped structure, but this outer wall thin, this layer outer side, for example, this is the guard cell, bean shaped, outer side thin, but inner side, if you observe, inner side very thick, like this. Inner side very thick, and this inner side, this is called, this is pore, opening pore. This is closing pore. Now this is closed type, this is open type. Open stomata and closed stomata. Right? So let's see the mechanism, how the stomata opened and when the stomata opened and when the stomata closed. Before that, the stomata in most of the plants are photoactive stomata. in xerophytic plants, photoactive stomata. What is photoactive and scotoactive? Photoactive, photoactive and scotoactive. Photo means light. That means if the stomata open during daytime, that is called photoactive. Scotoactive means if the guard cell open during night time, that is called scotoactive. Daytime photoactive, nighttime scotoactive. Right? Now let's see the opening and closing. There are several theories proposed to explain about stomata closing opening. But among these, the all scientists accepted the most important theory, K plus pump theory. K plus pump theory. K plus pump theory proposed by Leavitt. K plus pump theory. K plus pump. K plus pump. Leavitt proposed by K plus pump proposed by 1974, proposed by Leavitt scientist to explain the mechanism of opening and closing during photoactive, that is in photoactive stomata. According to this theory, accumulation of K plus ions into guard cell from subsidiary cell. I already told you these are subsidiary cells, sorry. Yes, subsidiary cells. From the subsidiary cells, K plus potassium, right here. Actually, they are deposited in the subsidiary cells. So, accumulation of K plus ion into guard cells. Now they are accumulated in guard cell. This is subsidiary cell, right? Subsidiary cells too, they are entered into the guard cell. Guard cell. If we draw only guard cell, surrounding K subsidiary cell, now the subsidiary K plus into the guard cell, right? It is carried out in the presence of light. In this situation, this coupled with efflux of protons leads to increase in pH of the guard cells. Accumulation of K plus ion into guard cell is associated with passive influx of Cl minus ion. Actually, along with the while the K plus enter into the guard cell, apart from the K plus, Cl minus also, chloride also enter into the guard cell. That means K plus with Cl minus enter influx into the guard cell, right? What happen if the Cl minus increasing inside the guard cell? Here the Cl minus ion gradually increased. Already inside the guard cell, apart from the Cl minus, malate also present. Malate anion. This is also negative. Malate negativity, chloride negativity, right? So the which value increasing in guard cell during daytime? Hello?
K plus, sir.
Here Cl negativity or positivity inside the guard cell?
Positivity.
There are how many K plus in guard cell?
One,
How many
sir.
K plus? How many K plus in guard cell?
One.
Only one. How Cl minus? For example, Cl minus. Cl minus also one, right? What about malate?
Yes. Also one, sir.
Malate. So here actually the number of malates are already accumulated inside the guard cell during daytime. Apart from the malate, already the malate anion existed. the extra anion in the form of Cl minus entered into the guard cell. As a result, which is increasing, positivity increasing or negativity increasing inside the guard cell? Which value increased? Hello?
Negativity, sir.
Negativity increased. That means already malate is there. Malate automatically anion, negative. The extra added Cl minus along with K plus ion, right?
Yes.
So here automatically the negativity value increasing inside the guard cell. Negativity means the water potential value negative, right?
Yes, sir.
So according to water potential, water movement from where to where? High water potential to low or low water potential to high? Already we discussed yesterday. Please respond. Doesn't matter whether it is right or wrong. What is water potential? Hello? What is water potential?
Increases the potential, sir, in the guard cells.
What increasing in guard cell?
Osmotic potential, sir.
When compared with substitute cell, right? Hello?
Yes,
Don't
sir.
get confused. Simply remember one thing. There are two cells, right? First one, guard cell, surrounded cell, subsidiary cell. Okay. Where the negative present in guard cell or subsidiary cell? Tell me first. Guard cell or subsidiary cell? Hello? Hello?
Sir, here the osmotic potential is more negative, sir.
Where more negative? In guard cell?
When they increase the ions of K plus, sir.
I am asking where the negative in guard cell or subsidiary cell?
In guard cell, sir.
Right, in guard cell, negativity increasing.
Yeah.
That means water potential low. Negative means low or high?
Low.
Low. What about subsidiary cell?
Low.
Is there any anions? Any negative value in subsidiary cell? Hello?
Yes, sir.
Any negativity in subsidiary cell? Actually, K plus, apart from the K plus and H plus sent out from the guard cell, H plus. So H plus only pluses present in subsidiary cell, but minus in the form of chloride and malate, negativity present in guard cell. So if we compare with both the cells, subsidiary and guard cell, as presence of H plus, high water potential in subsidiary cell, as presence of malate chloride, low water potential value in guard cells. So the water movement from where to where according to this? Did you attend yesterday class?
Yes, sir.
Then respond. What you learned yesterday? Any confusion with water potential? Any confusion with water movement based on water potential? Tell me the definition or tell me the movement of water based on water potential. High water potential to low or low water potential to high?
High to low, sir.
First write down on paper. Write down on paper. Water movement, high water potential to low, right? Write down, okay?
Yes, sir. Right.
Now see the diagram. See the screen. The screen, first subsidiary cell. What are present in subsidiary cell? Here, subsidiary cell only H plus. Subsidiary cell here. H plus. In guard cell, K plus, Cl minus, malate. Right? Okay. In guard cell, malate minus, Cl minus. Now tell me, where is high water potential? Subsidiary cell or guard cell?
Guard cell, sir.
Where is high water? High water potential means negative or positive?
Positive.
Where the positive in guard cells? Cl minus is positive. Malate is positive. Tell
No,
me,
sir,
Cl
they are
minus is
negative,
positive
no?
or negative?
Negative.
Malate positive or negative?
Negative.
Why did you tell positive in guard cell? What about subsidiary cell? Positive or negative? Subsidiary.
Subsidiary. Positive, sir.
Positive. Guard cell,
Negative.
where the high water potential? High water potential.
In subsidiary cell,
First of
sir.
all, let me complete. First of all, plus value high or minus value high?
Plus value, sir.
Where the plus value? Subsidiary or guard cell?
In guard cell, in subsidiary cell, sir.
Where the high water potential?
In subsidiary cells.
Where the low water potential?
Guard cells.
Now water movement
High to low.
from which cell to which cell?
Subsidiary cell to guard cells.
Right. From subsidiary cell to water movement into the guard cell. What if the water movement into the guard cell? Turbidity created in guard cell. That means due to the water flow, automatically these guard cells both are opened. Due to the pressure of water and turgor, due to the turgor pressure, stomata opened. Right?
Yes, sir.
Is it clear?
Yes, sir.
Any doubt in this? First stomata open. First mechanism, stomata open. Simple. Open stomata. Daytime. What happened in daytime? In daytime already malate. M for malate anion present. K plus from subsidiary enter into the guard cell. Apart from the K plus, Cl minus also enter. The same time H plus from guard cell to sent into the subsidiary cell. As a result, in subsidiary, due to the presence of H plus, high water potential, in guard cell due to the presence of malate and chloride, automatically low water potential. That's why based on water potential, water movement from subsidiary cell to guard cell. As a result, stomata open, guard cell open, right? This is done during daytime. So that's why stomata open. The same, how the stomata closed? How the stomata closed? Stomata closed in night time. So this is the guard cell. Is there light in guard cell?
No, sir.
No. Without light, can prepare the malate?
No.
No. So there is no malate anion, right? And also efflux, instead of influx, efflux is the K plus out. K plus out. At the same way, apart from the C plus, along with the K plus, Cl minus also sent back to the subsidiary cell. And H plus received. But in subsidiary cell, Cl minus concentrate increased gradually. Because already the subsidiary cell Cl minus present. Due to the efflux of the Cl minus out into the subsidiary cell, the concentration of Cl minus gradually increased. What about in guard cell? Now tell me, night time in guard cell, is there malate ion? Is there malate?
No, sir.
No malate. Is there chloride?
No, sir.
No. But only present of, what is presence?
H plus. K
But
plus
what about
SH plus.
right? What about subsidiary cell?
Subsidiary
cell Cl minus.
Yeah,
Cl minus
Cl
and
minus.
also malate anion also existed inside the. As a result, am I audible?
No, sir. In the middle, it was.
Okay. Right. Clear. So now compare with guard cell and subsidiary cell. Where is negative? Where is positive?
No, sir, in subsidiary cells.
Subsidiary cell positive or negative?
Negative, sir.
Right. Subsidiary cell negative. What about guard cell?
Positive, sir. H plus.
Now tell me the water movement.
Here, the water movement is decreasing to increasing, sir. The low potential to high potential.
Water movement. Water movement.
Low to high, sir.
Low water potential to high water potential.
Yes, sir.
Sure. What did you write earlier? Water movement. Just open your notes.
Yes, sir. Water movement, high potential movement to low potential movement.
What did you say earlier?
High to low, sir.
Remember, water movement, high water potential to low water potential.
Yes, sir.
Right?
Yeah. So in this low water potential to high water potential, sir.
Water movement, high water potential to low water potential.
Yes, sir.
Clear?
Yes, sir.
Tell me water movement.
Sir, in Cl minus, it increases gradually, sir. Water movement. In plus, they decreases.
Cl minus negative or positive?
Negative, sir.
Negative means potential decrease or increase?
Increase.
What is positive?
Increase in-
What is negative value?
Increase in subsidiary cells, sir. Cl minus. It is increase in
I am
subsidiary
asking
cells.
about water potential value. If the Cl minus value increasing in subsidiary cell, what about water potentiality? Water potentiality value increase or decrease? Water potentiality value.
Decrease, sir.
Right. What about guard cells?
Increase, sir.
Water potential increasing. As
Yes, sir.
presence of K plus, water potential increasing, right?
Yes,
Now
sir.
tell me the water movement.
The low to high, sir.
What low, what high?
Water, sir. High potential. Low potential to high potential, sir. Water potential is decreasing, sir, in subsidiary cells,
Hmm.
and in
Water
guard cell
potential
it is
decrease.
increasing.
Okay.
Yeah, in decreasing. Yeah.
Water potential increasing guard cell. What is the definition? Write down on paper. Water
Yes, sir.
movement, high water potential plus write down.
Yes, sir. Water.
Water potential, high water potential. Inside bracket plus. To
Yes,
low
sir.
water potential minus.
Yes, sir.
Right? So
Yes,
now tell
sir.
me the water movement from where to where? High water potential to low or low water potential to high?
High water potential to low.
Say again.
High water potential to low water potential.
Right. Now come to the screen. In night time, where the high water potential, guard cell or subsidiary cell?
Subsidiary cells.
Subsidiary cell.
Yes, sir.
Use your brain first. Keep concentrate while I telling the class, while I teaching the class. Where is minus? Cl minus where?
In subsidiary cells, sir.
Means subsidiary cell Cl minus. Minus means? Minus means?
Decreasing the water potential.
Write down on paper. Decrease water potential in subsidiary. Right? Okay. What about in guard cell?
Guard cell increasing, sir.
Write down. Complete?
Yes, sir.
Now read it. Where high water potential? Where low water potential?
So low water potential in subsidiary cells.
Right.
High water potential in guard cells.
Now read the definition. Water movement.
Water potential is high plus to the low water potential minus.
Now tell me the movement of water in night time.
Movement of water is low to high, sir.
Which cell to which cell?
Subsidiary cells to guard cells.
కొంచెం బుర్ర ఉపయోగించండి. ఏం రాసుకున్నావు అక్కడ ఫస్ట్ పైన డెఫినేషన్? వాట్ డిడ్ యు రీడ్? వాట్ డిడ్ యు రైట్? వాట్ డిడ్ యు లిసన్? What is the definition? What you write on paper?
Sir, water potential high to low water potential. decreasing water potential in subsidiary cells and increasing water potential in guard cells.
Okay. The water movement, high water potential to low water potential, low to high.
Low to high, sir.
See on paper.
No, high to low, sir.
Now tell me. Movement of water.
High to low, sir.
Huh?
High to low.
From which cell to which cell?
Subsidiary cells to guard cells.
See on paper in subsidiary cell water potential value. High or low? Why are you getting so confused? You already write the definition, you write the mechanism of water flow, and also clearly write the where the negative, where the positive. You are unable to tell the answer, even if you see the book. So during night time, the water flow from guard cell to out subsidiary cell. As a loss of water, the guard cell shrank, as a result closed like this. Water guard cell closed during night time. During night time, the stomata opened. This is the mechanism closing and opening movement, right? And after transpiration, the second unit, mineral nutrition. Mineral nutrition. Right, in mineral nutrition, the first actually in 1860, Julius von Sachs, a German botanist, demonstrated the first time that plants called grown to maturity in a defined nutrient solution in complete absence of soil. Actually, the plant grow with support soil. From the soil, the nutrients also absorbed. But the first time Julius Sachs, without soil, can grow the plants. This technique called as hydroponics. This technique called as hydroponic. Let me share the diagram. Just wait. Are you able to see this diagram? Hello? Are you able to see? Hello? Right. So this is only the minerals. Only minerals. Water. Is there any soil? No soil, only the mineral waters. Water with nutrient mediums. They can absorb the plant and the plants can grow. This technique is called as hydroponics techniques. This technique called as hydroponics. Hydroponics that can grow the plants without soil. That is called hydroponics. And here the nutrients, there are essential, non-essential nutrients. Essential elements are 16. The 16 elements, some are macro and few are micro. Before that, the criteria for essential elements, the first one, the element must be absolutely necessary for supporting normal growth and reproduction. Second, the requirement of the element must be specific and not replaceable by another element. the element must be, next one, the element must be directly involved in the metabolism of the plant. These three criteria most important for essential element. Of course, there are number of elements, not only 16. More than 16 elements are there in soil. But among this, only essential element, some criteria, the essential element criteria must be absolutely necessarily for supporting normal growth and reproduction. It is involved for growth of plant body and reproduction of plant. And also it require for, and it is never replacement for another element, and it must be directly involved in metabolism. Two types of nutrients, macronutrients and micronutrients. Let's see some macronutrients. First, the macronutrients are here: nitrogen, change, right, macronutrients. Macronutrients are nitrogen, potassium, nitrogen, potassium, phosphorus, NPK, phosphorus, potassium, phosphorus, potassium, sulfur, calcium, 1, 2, 3, 4, 5, and another one magnesium, sulfur, magnesium, right. So these are the major macronutrients. Let's see one by one. First of all, nitrogen. This is the essential mineral nutrient element required by plants in the greatest amount. That's why it is macro. It is absorbed mainly nitrate, NO3 minus. So sometimes it is also taken as nitrite or ammonium. And nitrogen is required by all parts of plant, particularly the meristematic tissue and the metabolically active cells. And this is one of the major constituents of protein, nucleic acids, enzymes, vitamins, and hormones. Right? Some of the deficient, what happens if the loss of deficiency of nitrogen? Mainly this nitrogen main constitution part of protein, nucleic acid, enzymes, vitamins, and hormones. And also nitrogen present in chlorophyll also. Right? If deficiency of nitrogen, what happened? Can guess? Simple. The mainly it is present in protein and also chlorophyll. If loss of nitrogen, malformation of chlorophyll, as a result formation chlorosis. Chlorosis occur in loss of nitrogen. Right? NPK. Next, phosphorus. Phosphorus absorbed by plants from the soil in the form of phosphate ion, either H2PO4 minus or HPO2 minus, any. So mainly this phosphorus is constituent of the cell membrane, plasma membrane made up of phospholipids. In phospholipid, phosphate present. Certain proteins also present the phosphorus. Nucleic acids. Nucleic acid means such as DNA, RNA. Nitrogen also present in nucleic acid. Nitrogen also present along with phosphorus. DNA, RNA, nucleotides, phosphate chain also present. That's why in the nucleic acid phosphorus present and required for all phosphorylation reactions. The phosphorus mainly involved in all phosphorylations reactions. What happen if the phosphorus loss? The mainly the loss of phosphorus, it shows stunted growth plant, stunted growth. And next one, potassium. Potassium absorbed in the form of K plus ion, K plus, required in more abundant quantities in the meristematic tissue, buds, leaves, and root tips. Potassium helps in maintain the anion cation balance in cells and also involved in protein synthesis also. Protein synthesis also potassium involved. And just now we completed opening and closing of stomata. Which theory explained opening and closing stomata? K plus pump theory, potassium pump theory, Levitt potassium pump theory, and activation of enzymes and in the maintenance of turgidity of cells. These are all done by potassium, right? Next one, sulfur. The plants obtained sulfur in the form of sulfate. Sulfur is present in 2 amino acids, cysteine and methionine. The main constitutes several coenzymes. Sulfur present in several coenzymes and also vitamins, mainly thiamine, biotin, coenzyme A, and pyridoxine. Right? And next one, calcium. The plants absorb the calcium from the soil in the form of calcium ion. Calcium ion, the calcium is required by meristematic and differentiating tissue during cell division. It is used in the synthesis of cell wall. What is the cell wall chemical? Cell wall chemical, calcium magnesium pectate, right? That's why it is involved in the synthesis of cell wall. It is also needed during the formation of meiotic spindle fiber, right? Calcium is also an important element in the help of the photolysis of water during photosynthesis. What is, I will explain what is photolysis later during photosynthesis class. Right, next one, magnesium. Magnesium, it is observed by plants in the form of magnesium 2+, activated the enzymes of respiration and photosynthesis, and also involved in the synthesis of DNA, RNA. The magnesium involved in synthesis of DNA, RNA during replication process, during transcription process. Magnesium is constituents of the ring structural chlorophyll. Like nitrogen, magnesium also the structural part of the chlorophyll and also help in maintain the ribosome structure. Right? So as it is present in chlorophyll, what happen if the efficiency loss of chlorophyll? Same like nitrogen loss, here also chlorosis occur inside the leaf. So these are the macronutrient, right? And any other in deficiency symptom, magnesium, no, right? So next one, the micronutrients. Once again, the macronutrients are NPK, nitrogen, phosphorus, potassium, sulfur, calcium, magnesium. So these are the major macronutrients. And micronutrients are Micronutrients are micronutrients. Iron, manganese, molybdenum, zinc, copper, boron, chlorine, nickel. Iron. The plants obtain iron in the form of ferric ions. Ferric ions. Iron is required in the larger amount compared to another micronutrients. When compared with other micronutrients, of course, it's micronutrients, but it required larger amount. It is an important constituent of protein involved in the transfer of electron like pyridoxine and cytochrome, and activate catalase enzyme, and it essential for formation of chlorophyll. Essential formation of chlorophyll. So here the essential of chlorophyll, formation of chlorophyll or structural chlorophyll mainly involved nitrogen, magnesium and iron. That's why the deficiency symptoms also same. What happen if the loss of nitrogen, magnesium, which deficiency symptom appeared in plant body? Chlorosis appear. Like this, if the iron deficiency occur in plants, chlorosis occurs. Right? Next one. Manganese. Manganese ion observed in the form of manganese ion. It activates many enzymes involved in photosynthesis and respiration and nitrogen metabolism. The function of the manganese in the splitting of water. Like copper also involved in, calcium also involved in splitting of water. Right, copper also involved in splitting of water. And manganese also involved in splitting of water during photosynthesis. I will explain later in photosynthesis class. Right? And different symptoms of manganese. Most important different symptom of manganese here, what given? Manganese, right. Gray spot is that given? Right. So loss of manganese, it leads to chlorosis, first one, and in what? Gray speck. Gray speck. In votes. In votes, gray speck disease. In Botany, Mars spot. Mars spot disease in Botany. Remember, molybdenum, gray speck in votes, Mars spot in Botany. Deficiency of manganese. Remember, right? Zinc. Next one, zinc. Plant obtained the zinc as a Zn2 plus. It activates various enzymes like manganese. It is needed in synthesis of oxygen. Synthesis of oxygen it required. And deficiency symptoms of zinc. If the there is little leaf, due to the loss of zinc, little leaf formation, little leaf leads to formation of little leaf, shortened leaf, right? Hello? Next one, copper. Cupric ions in the form of cupric ions, copper in the form of cupric ion Cu2 plus. Essential for overall or metabolic activities. Like iron, it is associated with the certain enzymes involved in redox reactions and is reversibility oxidation from Cu plus to Cu2 plus. And it is structural component of electron carriers like cytochrome C, oxidase, mitochondria, and plastocyanin in chloroplast. I will explain later this mechanism. If deficiency of copper, whiptail disease, where the copper, I think whiptail or dieback, right? Exanthema, exanthema disease and dieback disease. Dieback disease. Exanthema disease in citrus and also dieback disease also formed in citrus. Right, both. Whiptail disease, I will tell you later. So mainly the copper, exanthema, dieback disease. Next one, boron. Boron observed in the as a BO3 3 minus. It is involved in pollen germination, cell elongation, cell differentiation, and carbohydrate translation. A deficiency of boron, is there some, where the boron? Hot rot of sugar beet. Deficiency of, just wait. Hot rot of sugar beet. Hot rot of sugar beet form loss of boron. Right. Next, molybdenum. Molybdenum in the form of MoO2 plus. It is component of several enzymes, including nitrogenase and nitrate reductase. The molybdenum deficiency, whiptail disease. Molybdenum. Next one, chlorine. It is observed in the form of chloride anions, Cl minus. Along with the Na plus and K plus, it helps in determining the solute concentration and anion cations balance in the cells. It is essential for water splitting reaction in photosynthesis, like manganese, like copper. Chlorine also involved in water splitting reaction. Last one, nickel. It is the recently reorganized in the 17th essential element nutrient as act an activator for UGS, an important enzyme nitrogen metabolism, and it is also found in induce the disease resistance in some plants. So these are the essential elements, macro and micro, and this question is very important in long questions, right? Hydroponics important in short question, right? Next one, enzymes. What are the enzymes? Enzymes are chemically proteins. Hello? Hello? Are you there? Hello? Right. Now, enzymes. Enzymes are chemically proteins. The study of enzymes called as enzymology. Almost all enzymes are protein, and there are some nucleic acids that behave like enzymes. There are called ribosomes. One can depict an enzyme B line diagram. An enzyme like any protein as a primary structure, such as amino acid sequence in the protein. An enzyme like any protein as a secondary and tertiary structure too, when you no need that. And these are the temperature sensitive, and each enzymes are temperature sensitive and heat sensitive. Let's see the properties of enzymes. Properties of enzyme we will complete in next class. Already 1 o'clock, right?